Welcome to Labii tutorial. Labii provides the services of electronic lab notebook and laboratory information management system. In this tutorial, I will demonstrate how to use the Plasmid editor widget to document, annotate, and visualize plasmids in DNA cloning. Please create a new record and add a widget Plasmid editor section to get started. If you are not sure how to do that, please watch the Labii user training video. By default, an example sequence is added. The widget comes with a menu bar on top. The menu bar includes all the functions necessary to modify the plasmid sequences and features. Each function is represented by an icon, and when your mouse is hovered over the icon, the tool's name is displayed. A menu bar can be seen only in edit mode, and hidden in read-only mode. The main body contains a linear view on the left, and a circular view on the right. On the bottom, the status message shows the meta message of the selections. Specifically, the start and end positions, the length of the selection, the TM and the GC. The right side of the status message shows the percentage of zoom. Various methods can be used to load your own sequences. You can drag and drop files into the cloud icon. Alternatively, you can click the cloud icon to select a support file. You can also select a file from the menu by clicking Open. Data can also be loaded directly from an NCBI accession number, or an IDIMPART number. To do so, click the Load from Accession button, select an accession, and click the Submit button. Views can be switched by clicking the Views icon on the menu bar. Both views display both the linear and circular viewer. In the linear viewer, DNA sequences are displayed along with features and axes. You can zoom out to view only features. In the circular view, you can see plasmid maps. Feature views show a list of features that have been annotated to plasmid sequences. Other tools in the menu bar can be used to add, edit, or remove features. On the Enzymes view, you can see the digest enzymes used, and their cut sites. The Add Enzymes tool can be used to add, edit, or remove enzymes. You can search for sequences within plasmids. The search function allows you to find the position of a given sequence. A primer can be added to a plasmid by searching for its position and clicking on Add Feature. Click on Search on the menu bar. Enter a query in the query field. You can use the wildcards below. Provide the maximum allowable mismatch between the query and a match. Then click Submit. The search results will be highlighted in yellow. The plasmid can be renamed by clicking the Rename icon on the menu bar, and providing a new name. The new name will be updated immediately on the map. Plasmid Editor allows the modification of plasmid sequences. To insert bases, click the position you wish to insert in the linear viewer, and then click Insert Bases at Selection. Type or paste the nucleotides into the pop-out modal. Press Submit to submit. The bases can be modified by selecting the sequences, and clicking Edit Selection Bases in the menu bar. This will display the selected sequences in the pop-up modal. Make any changes and click Submit. To delete selection bases, select the sequences and click Delete Selection Bases. You can copy selected bases by selecting the sequences, and clicking Copy Selection Bases. You can also add, edit, and delete features. In the menu bar, click Add Feature and provide the name, type, start position, end position, direction, and color. When you're done, click Submit. In addition, you can also select a feature in the linear view and click Add Feature. The start and end positions will be automatically filled in the feature form. If you wish to edit a feature, select it and click Edit Feature. Edit is necessary, then click Submit. You can delete a feature by selecting it and clicking Delete Feature. Specific enzymes can be added to see the digest results. To do this, click the Add Enzymes icon, select one or more enzymes in the Enzymes field, and click Submit. You will then see the enzymes in both the linear and circular views. You can also see all the cut sites of the enzymes in the enzyme view. The menu bar also allows you to zoom the sequences, change the case, and hide or show complementary sequences and axes. I hope this video is helpful. Please subscribe to our channel to get the most recent updates. If you have any other questions, please submit a support ticket, or schedule a meeting.